A very good morning to everybody. So, today in this concluding class, we will just basically finish off the remaining part on nonmetals. And this is basically what we are continuing the fourth part of the present series. So, in this particular one, the remaining one, what we are talking about, how iodide is assimilated, which is a very well known thing and which is basically the common knowledge to everybody, that this particular one, how it is getting inserted into the enzyme system or the hormone system. So, next we will see also, after finishing this remaining part on iodine and thyroid thing, we will just basically another interesting element, which is selenium. So, selenium, how it is different. So, along the periodic table, how we can compare this with the two other members of this group. So, we have sulfur, selenium and tellurium, and so far we know much of these developments have already been taken place for the corresponding bioinorganic part or the biological chemistry related to the nonmetals, where this particular sulfur atom from the cysteine, so it can be present in the amino acids like cysteine, cysteine and methionine, but in this particular case these are functioning as simple donor atoms to different metal ions, say iron. So, iron in +2 or +3 when they bind to the sulfur. So, basically this particular part is not a truly inorganic part related to the nonmetal chemistry to the biology, but it is something where the sulfur is present and which is bound to the iron center. So, most of the cases it is the hybrid thing where the properties of the donor atoms as well as the properties of the metal centers can take part in controlling all these biological reactions, and compared to that the nature of the selenium atom is relatively different compared to sulfur, and when selenium is taking control of all these things. So, we will not talk in terms of its corresponding binding or coordination to any such metal center like iron in the ferrous or the ferric state, but the individual thing, that means whether this selenium atom, how it can control several biological reactions. So, first we will just go back to the remaining part of this nonmetals in biology in part 4, where we are talking about the T4 and T3 hormones. So, which is the tetraiodoform and the triiodoform, and already we have seen that they are generated in the thyroid gland and can be circulated throughout the body and regulates this is to regulate a wide variety of metabolic processes. So, they basically require for some metabolic processes. So, if we just see that our body metabolism, that means we know the very simple thing, that is the BMR, which is very important, the basal metabolic rate, how we go for the different type of metabolic rate for the anabolism and the catabolism. So, metabolism is definitely dependent on these two hormones. So, it is the T4 initially formed, whose life is very high, and the active one, the derived one from the T4, which is the T3. So, it can control the metabolism. So, it is very much dependent on, not even for the deficiency related disease like goiter, but it can also hamper some of our very important and very basic metabolic reaction pathways. So, if our metabolism is getting affected, so we can consider that not only for the expression of this iodine deficiency in terms of the disease goiter, but also the metabolism pathways can also be hampered. So, if we can have some defective metabolic pathways or the defective or the different BMR rate what we expect, then we can control the corresponding role of the iodine as well as the iodide ion in all these biochemical processes. So, these are very important for finding the corresponding basal rate of metabolism. So, how the rate of metabolism is getting affected, it is due to the corresponding concentration and the interaction of these T4 and T3 molecules for the metabolic pathways. It is also important for regulating our body temperature, because these temperature dependent reactions, the rate of the reaction all we know from the thermodynamic and kinetic point of view. We know that the rate of the reaction is getting enhanced when the body temperature is increasing or the reaction temperature is increasing. Similarly, when all these biochemical reactions are taking part in our system, and if we can go off a temperature state from one to other, so for 1 degree or 2 degree rise in temperature, the corresponding rate of the reaction or this rate of metabolism reaction in fact is also getting changed. So, for the temperature regulation as well as the rate control for the metabolic pathways are also dependent on iodine, and it can also be important for expressing their role in the synthesis of different protein molecules. So, protein molecules are also dependent on the corresponding deficiency of iodine and the corresponding related hormone molecules for their synthesis. So, what we see that when we have the reaction, we basically bring something, that means what we know all, the peroxidase activity, which is very important. So, whenever we have something, that means the peroxidase activity is taking place, that means we are just handling this O2 2 minus ion, the peroxide ion, and we all know that during our respiration and all we consume this dioxygen molecule, which is very important for our very survival and which initially can be taken up by the corresponding myoglobin and the hemoglobin molecule. The heme proteins, they are basically responsible for binding of this O2, and in some processes if we can transfer electrons. So, if we can transfer more than one electron, so electron 1 and electron 2, if we can inject into the antibonding orbitals of the O2, that means if we have the different molecular orbitals of the O2 molecule, and if we can feed these electrons to those antibonding orbitals, we will find that we can convert it to the peroxide ion. So, peroxidases are also there. So, if we can consider this peroxidase activity, which can attack on the generation of this peroxide ion, and we can responsible for the destruction of the peroxide ion, because the accumulation of peroxide ion in any living organisms, in our body, in animals body, in humans body, are very important, because they are pretty strong oxidizing agents. So, these are oxidizing in nature. So, if we want to get rid of the accumulation of peroxide ion, that means the concentration, their concentration is important. So, if we want to go for the destruction of the peroxide ion, we always take the help of the peroxidases, but the standard peroxidases what we can have, which basically these are based on iron porphyrin macrocycle. So, iron porphyrin macrocycle is there, that means the heme proteins are there, and heme proteins are responsible for both peroxidase and the catalase activity. So, this iron centered peroxidase activity was known to us, and we have studied earlier for this peroxidase activity, but this particular activity is very much required for the destruction of peroxide ion. So, it is a very important pathway for the destruction of this peroxide ion, and if that particular destruction we can utilize for some useful function, that means not only the removal path, the removal of toxic peroxides from the system, from the living organisms is important, and at the same time if we are able to see some peroxidase activity for the synthesis of some good molecule to our system. So, if we are able to synthesize some good molecule at the expense of peroxide ion. So, we will find that some molecules. So, in this particular case, if we are able to make the hormone, the T3 and T4 molecules, if we are able to make with the help of this peroxide. So, what we take, we just basically take the iodide ion. Iodide ion through its reaction with hydrogen peroxide is basically liberating or in situ generating the iodine molecule, and sometime like our laboratory processes also, that this iodide oxidation while producing the iodine molecule can also be trapped within this particular species, forming triiodide anion. So, all these things will be in equilibrium concentrations, the relative ratios will be there for the iodide, iodine and triiodide depending upon the individual concentration of all these species, and depending on the starting iodide concentration into the medium, how much iodide we are getting through the food material or through the salt intake what we are utilizing for our intake along with that of our sodium chloride. So, this particular peroxidase activity can be very useful if we just relate it to the thyroid gland. So, in thyroid gland, if we can have the corresponding molecule which is available, which is readily going for the corresponding oxidation of I minus to IT, we get the corresponding molecule as thyroperoxidase. So, the thyroid gland which bears and which shows the corresponding peroxidase activity is the corresponding thyroperoxidase. Thyroperoxidase. So, this particular thyroperoxidase, so thyroperoxidase is there, so which is TPO, the thyroperoxidase. So, any such conversion, so if we have the amino acid on the left and we have iodine as iodide ion, so we can go for the corresponding iodination reaction. So, iodination reaction can take place with the help of this TPO molecule. So, the generation of these T4 and T3 molecules are therefore dependent on the availability of these thyroperoxidases. So, when we see that the enzyme which is available now to us is the thyroperoxidase, and we have to attach the iodine to the tyrosine residue of the long amino acid based protein chain or the polypeptide chain which is present in thyroglobulin molecules in thyroid gland. So, the iodination can take place if the corresponding phenol ring of the tyrosine amino acid is going for the corresponding iodination reaction at one position, we get monoiodotyrosine molecule, and if it goes for the corresponding substitution of hydrogen atoms by two iodine atoms on the ortho positions of the hydroxy group of the phenol ring, we get the diiodotyrosine molecule. So, the effect of these reactions for the thyroperoxidase on iodine or iodide ion is basically generating the MIT and DIT molecules. So, we just produce the MIT and DIT molecules to us, and this is the corresponding monoiodidation reaction on the tyrosine residue, and if we can go for this particular substitution also, we get the corresponding diiodination molecule. So, tyrosine is basically now is giving for the MIT molecule and the DIT molecule. So, these MIT and DIT molecules in turn can go for the synthesis of the T4 and T3 molecules in the reaction medium. So, when we have these DIT units and two such DIT units, when they couple together produces thyroxine. So, thyroxine molecule, what we know that it has basically four positions which are occupied. So, it is a bisphenol unit. Thyroxine, as we have discussed in our previous class, that it is a bisphenol unit. So, we will have 4 phenol positions which are ortho to the OH group or the O group as thioether. So, all these 4 positions can be iodinated to produce the T4 molecule, and when we combine one MIT with one DIT, it can produce the T3 molecule, which is much more effective molecule in terms of the corresponding thyroid activity compared to the corresponding T4 analog, because we always get the T3 molecule from T4 through deiodination reaction. So, there also we should take the help of deiodinases, which is selenium based molecule that we will discuss in this class today only. So, this particular cases we just get then some proteases which are responsible for the hydrolysis of the long protein molecules. So, they digest the iodinated thyroglobulin. So, thyroglobulin is getting iodinated through the use of thyroperoxidase molecule, and this iodinated thyroglobulin molecule, when we have the corresponding formation of the bisphenol unit of the tyrosine residue, we have already formed T4 and T3 units through coupling of 2 DIT and 1 DIT with 1 MIT. So, both T4 and T3 molecules are already formed there and already present in the thyroglobulin molecule as colloid, and these two both are biologically important molecules, biologically important agents to central metabolic regulation. That means, it can control the corresponding metabolic pathways and the metabolic rate also. So, when they are formed inside within the thyroglobulin, they can be eliminated to the bloodstream. So, this is the second molecule, which is the DIT molecule. So, the previous one was, this is the monoiodinated one, and the second one is basically the diiodinated product. So, when they are forming, so inside the thyroglobulin molecule they go for the corresponding condensation. So, what we see is a very simple reaction like that of our phenol, is a pendant phenol unit only, because this is the glycine part. So, glycine part which is the amino acid part. So, amino acid part which is substituted the para position of the phenol. So, these two groups are basically responsible for the iodination reaction, the simple iodination reaction of the phenol unit. The way we were discussing that the bromination reaction also, if we can have the corresponding mixture of bromide and bromate or the elemental bromine, we can go for the corresponding bromination reaction of simple phenol unit in the laboratory to give us the corresponding tribromophenol or picric acid. So, this DIT molecule can bind with another DIT molecule, giving us the T4 molecule. So, the routes are now well known and well established. So, these DIT molecules are also known as the modulator of thyroid peroxidase, because they are basically combined with itself or another MIT molecule in the colloid of the thyroid follicle, and triiodothyronine is formed when this DIT molecule attaching with another MIT molecule. So, once these are formed, and then only we can go for the corresponding coupling. So, this is basically some kind of very simple and well known reaction for this is the substituted one. So, if you have the phenol, so formation of T3 and T4 is mainly responsible for the formation of the corresponding bisphenol unit. Like in the polymer chemistry, what we see that it is possible to go for the corresponding bisphenol molecule. So, this is basically the simple backbone where we have the amino acid fragment at the para position. We can have the positions for iodination. These are the positions for the different iodinations. So, this entire molecule what we get is the corresponding bisphenol molecule. So, basically we have like the polymer formation, we have the corresponding bisphenol unit, and the formation of this bisphenol unit is interestingly is taking place in our typical body in our some active gland, which is the corresponding thyroid gland. So, thyroid gland is therefore responsible for this particular phenol-phenol coupling reaction to give us the bisphenol unit. So, this phenol-phenol coupling reaction is taking place when we have the iodinated form on this is the amino acid substituted part. It is not that the bisphenol is forming, then we go for the iodination. So, the step for iodination is well known and well established now. So, the corresponding steps for iodination reaction is now known. This is therefore known to us. So, what we get if we just consider the entire picture, that we have some iodide ion which we are taking as a salt or as the food material, and for this we want to produce the corresponding iodine molecule, and this iodine molecule is produced only with thyroperoxidase activity. So, this thyroperoxidase activity is giving us, because we have some accumulated hydrogen peroxide in the system. So, always we depend on some amount of hydrogen peroxides, and in some way it is also beneficial to us also. It is showing the corresponding peroxidase activity, destroying the accumulated concentration of hydrogen peroxide in the living organisms. So, we have tyrosine and iodine is there. So, this can give rise to two such molecules, that diiodotyrosine and monoiodotyrosine. So, these are completely different from the T3 and T4 molecules. That the first step is typically the tyrosine molecule is iodinated, and when we go for the binding of these two DIT molecules, we get the T4 molecule, and when one DIT and one MIT is attacking this, so when these two, basically the MIT and DIT molecule, the monoiodotyrosine and diiodotyrosine are reacting to each other, we get T3 directly. But that also can be controlled by the living organisms, but the most stable species, what we have seen, what we are discussing from our previous class, that T4 is the most stable thing, and when the concentration of these DITs are higher compared to MITs, because the formation of T3 is dependent on the formation of the corresponding amount of MITs in the system. But if the concentration of these MITs are less compared to the DITs, we will only go for the corresponding formation of the T4 species. And T3 is more active. So, this T4 can we consider sometime as the pro-hormone. So, pro-thyroid hormone, so which can be activated if we know that the corresponding T3 through one step deiodination reaction can give rise to T3, and T3 is much more active than T4, then selenium containing enzyme, which is deiodinase. So, deiodinase is there operating on T4 to giving rise to the required amount of T3 molecules. So, we are basically handling on some of these molecules where we are talking in terms of two peroxidases. So, apart from the iron bearing peroxidases also, so studies on iodine in biology. So, what we are studying so far is the corresponding reaction of this iodine in biology, and we find that not only we are studying the corresponding incorporation of iodine in the biological molecule, but also we are talking some amount of its corresponding peroxidase activities. So, when we have the TPO molecule, the thyroperoxidase, we see that the corresponding TPO is responsible for its corresponding enzymatic reaction for the generation of MIT and DIT molecules. And in other case, because we see that selenium bearing peroxidase is also there, which is responsible for another interesting amino acid based species, which is glutathione. So, sulfur oxidation can take place. So, we can have the corresponding glutathione peroxidase. Since we are talking in terms of the corresponding peroxidase activity, we see that whenever we have this H2O2, the peroxidases can operate or act on this hydrogen peroxide to form water molecules. So, if it is thyroperoxidase, which is utilized for the hormone production, and if it is glutathione peroxidase, it will basically go for the destruction of the corresponding simple peroxide molecules. So, both these peroxidases are basically working or destroying the corresponding accumulation of hydrogen peroxides H2O2 within the system. So, this TPO is also known as thyroperoxidase. So, thyroid peroxidase or thyroperoxidase therefore is an enzyme. It is expressed mainly in the thyroid and liberates iodine, what we have seen just now, for the addition on to tyrosine residue on thyroglobulin for the production of thyroxine and triiodothyronine. That means the two thyroid hormones, the T3 and T4, and formation of their individual components, that means the formation of T3 and T4 are typically dependent on the corresponding TPO molecules, that means the thyroperoxidase molecule. And sometimes this TPO molecule is also stimulated by TSH, the thyroid stimulating hormone, one such hormone is also there, that thyroid stimulating hormone, which is basically activating the TPO, and TPO is in turn can act on the iodide ion for the production of iodine for the thyroxine synthesis. And this particular upregulates the corresponding gene expression. So, TPO has some interrelationship with that of thyroid stimulating hormone TSH for its production and for the corresponding gene expression, such that we can produce the required quantities or required amount of TPO as well as TSH molecules. So, we see that it sometimes is required, that means if we can have something, that means our thyroid gland is proactive, its activity is more, and which is producing more amount of the corresponding thyroid hormones, the thyroxine and triiodothyronine. So, if the production of T3 and T4, as we have seen, it can control the corresponding metabolic rate of our body. So, if the corresponding production, if they are producing in higher concentration, that means the gland, the thyroid gland, we have, and if the thyroid gland is proactive, its activity is more, and both these two, that means the T3 and T4 concentration is high. So, what in that particular case what we can do, because the production of hormone in our system, in our body should be balanced, it must have some optimum concentration. If the concentration is less, we have some symptom, some corresponding disease symptom, and if the concentration is high, we can have another type of diseased condition. So, both are not good for our system, that means high concentration of T3 and T4 and the low concentration of T3 and T4. We must have some optimum concentration for all these molecules. So, if it is proactive and it is producing more and more the corresponding T3 and T4 molecules, so what we can do, we just can go for the reducing the activity of this thyroid gland. So, how to reduce the corresponding activity of the thyroid gland, such that we want to produce less amount of T3 and T4 molecule in our bloodstream. So, if we know the entire mechanism of formation of this corresponding T3 and T4 molecule, and which particular molecule is very important for the synthesis and how the enzymatic reaction is taking place. So, for us it is therefore a very important pathway that we can control the corresponding enzymatic path. That means, the enzyme which is there and which is available for the production of T3 and T4 molecule should be inhibited. That means, if we can go for the corresponding inhibition of the enzyme activity, we can reduce the corresponding production of T3 and T4 molecule. For the typical iodination reaction on these 4 positions, we see that the corresponding tyrosine amino acid can be iodinated, and we can go for the corresponding thyroperoxidase TPO, and this TPO is acting on this corresponding tyrosine residue, and this particular. So, if we go for something, the mechanism where we can reduce the activity of these TPO only. So, how to slow down the corresponding reaction for this TPO molecule? We can have some mechanism for that, and we can go for the corresponding inhibition reaction. So, the inhibition of TPO is therefore can be done by the use of some thioamide drugs. So, we can have some position where these groups are available, and these thioamide drugs can be considered as the corresponding binding of this TPO unit. And what are those thioamide drugs? These are basically the corresponding propyl thiouracil and methimazole units. So, these are very small molecules having some thioamide backbone. So, the enzyme inhibition can be taken place if we can have, we all know that amide function is CONH. This is our typical amide function, and thiamide will have the sulfur group, because this oxygen can not react so nicely with all these enzymes active sites, but the sulfur can interact and bind inside the corresponding enzyme active site. So, this thiamide type of molecules, it can be a small ring molecule also. So, thiamide types of molecules can be utilized for the corresponding high activity of the thyroid gland, and those can be used as the corresponding drug molecule. So, they are basically good inhibitor molecule. So, thiamide type of designing is important for inhibiting the corresponding TPO activity. So, what is that particular molecule? So, one such example is that this is the corresponding molecule where we have the C double bond SNH function. So, this particular part is the thiamide part, and they basically go inside the corresponding enzyme, the TPO enzyme, and basically what happens that this particular sulfur is a very good unit. It can go for some hydrogen bonding interaction, or it can bind to some other group. So, through sulfur it is starting interacting with that, and through this NH, because this NH can also be a very good hydrogen bonding donor. So, if within the enzyme structure some hydrogen bonding acceptor is present, so this hydrogen can go for interacting that particular site through hydrogen bonding. So, both sulfur and this NH function can go and interact with that of our TPO molecule, and it is no longer available for its activity for iodide oxidation. So, Thyroperoxidase activity can be inhibited by this sort of small molecule, which is known as the corresponding MMI type, where the basic backbone should have always the thioamide group. So, these thioamide drugs are therefore known as a very important type of drug molecule, which is known as anti-thyroid drug. These are known as anti-thyroid drug. So, if we want to reduce the corresponding activity of the thyroid gland for the production of thyroxine and triiodothyronine, we should have some thiamide based drugs which are known as the corresponding anti-thyroid drug. So, one is known as methimazole. This is the typical one which is known as the methimazole, is also known as tapazole or thiamazole. So, sulfur bearing imidazole type of ring is, because this the nitrogen bearing 5-membered ring. Without this sulfur, we know that these are known as imidazole. And if the C double bond S group is present between these two nitrogen atoms, we can consider these as thiamazole. So, thiamazole is the corresponding drug, and this drug can inhibit the corresponding TPO activity. Next, we will just go for the remaining part before we just conclude this class for all this activity for the metal ion and activity for the non-metal centers also. So, in this particular case we will find that how we can go for the involvement of selenium in the biological system. So, selenium biology. So far we have seen for the different deiodinase reactions and all other reactions that we, in case of the corresponding production of T3 from T4, we have seen in our previous class, that means we can go for deiodinase activity, and that deiodinase activity is basically responsible for the presence of selenium in the group. So, when selenium is present over there and if it reacts with T4 plus proton, it basically go for the production of T3. So, deiodinase basically is a selenium bearing molecules, that is why it is very interesting group of molecules which are present there. So, we should have selenium in our food intake. Selenium is therefore essential micronutrient for our body for the right production of the T3. So, the formation of T3 is dependent on iodine, formation of T3 is dependent on TPO molecule, and also this formation of T3 is dependent on the selenium. So, if selenium groups are not present that much, we can go for the shortage of the corresponding deiodinase molecule and we can not get the corresponding right amount of T3 from the system. So, deiodinase we can have the selenium, and which basically is trapping the corresponding iodide group for the corresponding redox reaction for the formation of T3 from T4. So, this particular one therefore can be considered as a very useful micronutrient for animals, for human being. In some plants it is basically occurs in amounts which are toxic as forage or locoweed, but it is a very important component for the amino acids selenocysteine and selenomethionine. We know the cysteine molecule, we know the methionine molecule, and the cysteine molecule and the methionine molecule, when they are attaching to the corresponding selenium atom, we get the corresponding selenocysteine and the selenomethionine molecule. And we have been discussing very much about the corresponding other peroxidases, that means the TPO, and we have also discussed that the corresponding consumption of hydrogen peroxide by glutathione peroxidase. But in our body, this selenium what we can have as trace element from the food material mostly, and it functions as cofactor for glutathione peroxidase, that means selenium is present in glutathione peroxidase and in certain form of thyroredoxin reductase. This is a redox reaction like rubidoxin and ferridoxin molecule, and is sulfur bearing redox reactions can take place. So, is a very group of molecules are known as thyroredoxin reductase molecule. So, selenium what we can have, selenium containing proteins we all get also can be produced from inorganic selenium salts like that of sulfur bearing salts through the involvement of selenophosphate. So, we see that if we have the phosphate groups we all know, which is PO4 3 minus, and if we are able to go for the substitution of one oxygen by sulfur, we get the corresponding thiophosphate groups for the inorganic one also, because if we all know that P4O10, the corresponding phosphorus pentoxide, dimeric form of phosphorus pentoxide is there. Similarly, P4S10 is also possible, that means oxygen atoms can very nicely be substituted by sulfur groups. Here we have phosphorus oxygen bonds, here we have phosphorus sulfur bonds. So, in the same fashion, if we are able to go for the substitution of one of the oxygen atom by selenium, we get the corresponding selenophosphate. That means, we have phosphorus attached to selenium and one oxygen less, that means the 3 oxygen atom, but the overall charge on the species would be 3 minus still. So, therefore, we see that in this particular case in the selenophosphates we have the relevant phosphorus selenium bond. Same is happening in the case of the formation of selenocysteine and selenomethionine molecule. So, as we have seen earlier, as we mentioned in our previous class also, that in deiodinases selenium is present for the removal of iodine in this DIO molecule. In this particular DIO molecule selenium is present, which is taking up this iodine. So, it basically playing the corresponding functional role in the thyroid gland, otherwise we will not be able to produce the right amount of T3 molecules from the T4. It participates as a cofactor for the 3 thyroid hormone deiodinases. So, there are 3 types of deiodinases which are responsible for the production of right amount of T3 from the T4 molecules. These enzymes activate then deactivate the various thyroid hormones and their metabolites. So, with these enzymes basically first we go for the iodination reaction, and for the deiodination reaction we use the corresponding deiodinases, and sometimes it may inhibit the Hashimoto's disease, in which the body's own thyroid cells are attacked as alien. That if we have the thyroid cell, and we all know that the thyroid cells are utilized for the production of the corresponding amount of thyroxine molecules, the hormone molecules, but in some cases this particular one, that means this particular thyroid cell can be taken as the alien, that means it is unknown to them, unknown to the system. So, a reduction of 21% of TPO antibodies, which are the antibodies of the corresponding peroxidase, is reported, and in that condition is also is not a good condition, which is a diseased condition. We should have some dietary intakes, that means through our food material in the form of food or in some other conditions as salt also, we can take at least 0.2 milligram of selenium in our system. So, this particular selenium, presence of this selenium, which is very important for this non-biological part as well as the biological part. So, the first and foremost corresponding selenoenzyme which has been identified is the corresponding glutathione peroxidase. So, which is operating on the glutathione molecule, which is a very important antioxidant molecule. So, if we have the first enzyme which is glutathione peroxidase, so it can show the corresponding peroxidase activity. So, this particular peroxidase activity, we will see that the deiodinase activity is dependent on the corresponding presence of selenium. So, peroxide activity is also use the corresponding involvement of the corresponding selenium group. So, this GPX, the glutathione peroxidase, so is the glutathione peroxidase is operating on peroxide. So, it is the corresponding activity is the peroxidase activity, which is also bearing the selenium atom. So, when we have the selenium atom, this is incorporated within the protein chain. So, which is basically incorporated within the protein chain and in the form of selenocysteine residue. So, is if we have the corresponding cysteine residue, so this is within the protein, how it is going to incorporate in the protein chain as selenocysteine. So, like cysteine, which we abbreviate as CYS, we abbreviate this as it is selenium and cysteine sec. So, we have the selenocysteine residues in the protein chain like that of our cysteine residues. So, that selenocysteine residues are responsible for the corresponding catalytic redox cycle. So, basically is a redox reaction, that means the oxidation and reduction can take place. So, we can have the corresponding catalytic redox cycle involving glutathione and the glutathione reductase. So, it is basically oxidized in this particular form. So, when this sulfur bearing compound, the corresponding glutathione is present, is basically in the reduced form it is giving the GSH, and in the oxidized form it is the GSSG form. And when these are basically coupled with that of our glutathione peroxidase bearing selenium group, which is acting on the organic alkyl peroxides in presence of proton, forming the corresponding ROH group, and our GPX is forming a COH, that means the selenol is forming. So, this selenol is forming over there and the corresponding selenic acid is also there, and this basically going for the corresponding GPX, then the selenium group bearing thing, that means is S minus. So, when it basically goes for the corresponding reaction with this GSH, so is if we have the GSH is there, which is coming from this side, and this GSH. So, GSH is go and attack to this particular selenium bearing glutathione peroxidase. So, is forming the S and G group. So, what we see that in this case is, that means the corresponding formation of these sulfide bond, that means the disulfide unit, and we all know how these disulfide units are forming. These disulfide units are basically forming through oxidation of these thiol groups. So, if you have SH unit which can be oxidized to SS unit, the redox equivalent can be stored inside the molecule in the same way that the glutathione peroxidase is basically acting. That means, inside the molecule itself it can go for the corresponding transfer for this sort of SS formation with the use of NADP and GR, which is glutathione reductase. So, this peroxidase activity of these is dependent on the involvement of GR also. GR is our glutathione reductase, and we get all these things. So, this particular formation of this selenic acid and the corresponding hydrogen peroxide is getting destroyed through the production of water molecule and through the consumption of these organic hydroperoxides, and the anionic form is again regenerated through this particular cycle. So, is basically working on the corresponding glutathione molecule and is basically consuming the corresponding hydrogen peroxide or the different organic hydroperoxides or organic this one, this organic hydroperoxide is basically consumed. So, this particular reaction, because is a first of this type of enzyme which bears the selenium atom and first discovered. So, that is why the glutathione peroxidase molecules are so important to know. And the second one, we have already seen the corresponding deiodinase molecules, and the third one which is also known, which is the corresponding thyroredoxin reductase, which is the thyroredoxin reductase molecule. So, this particular molecule basically go for this corresponding, this is the glutathione peroxidase behavior what we have seen, and that organoselenium compound L-besen is also utilized as drug for the corresponding action of this glutathione peroxidase, and the corresponding catalyst is utilized for the generation of this or destruction of this hydrogen peroxide. So, this thyroredoxin reductase, which is basically another the third type of selenium enzyme, which generates the reduced thyroredoxin from the dithiol, again we get the dithiol and disulfide thing, and this particular molecule is very much important for reducing the ribonucleotide reductase, which we all know that is basically a iron bearing enzyme, but how we reduce the ribonucleotide reductase is now known as a selenium dependent procedure, where the ribonucleotide reductases are reduced for the generation of the corresponding RNA molecules, the DNA precursors from the RNA precursors. So, from the ribonucleotide reductases go for the deoxyribonucleotides, and the deoxyribonucleotides are incorporated to the corresponding DNA molecules. So, all these three types of molecules what we have seen throughout this particular course, that we have started our journey from the involvement of the different metal centers, the metal ions, in there. When we have the metal ions, we have the corresponding chance for getting these metal ions in the different oxidation states, whether it is Mn plus and in plus 2 or plus 3, and it is the typical environment. But when we are talking in the last few classes on nonmetals, it is the corresponding nonmetal center like that of our iodine as iodide or selenium as Ac minus through some terminal units, and that they are further interacting with some other groups which we cannot get from the corresponding donor atoms, like the corresponding sulfur base system what we can have as the donor groups for these metal bearing metalloenzymes or the corresponding molecules, where the metal is bound to these particular sulfur groups as the corresponding donor groups. Thank you very much.